Welcome to today's lecture on cell biology, specifically focusing on the intricacies of renal function and various aspects of renal cell biology. The kidneys play a crucial role in maintaining homeostasis by regulating fluid balance, electrolyte concentrations, and waste removal. In this lecture, we will explore 12 key topics that will provide a comprehensive understanding of the cellular mechanisms underlying renal function and dysfunction. So, let's dive in and explore the fascinating world of renal cell biology. Renal cell apoptosis refers to the controlled process of cell death that occurs in the kidneys. While apoptosis is a normal physiological process involved in tissue turnover, it can be dysregulated in various renal pathologies. Several factors regulate renal cell apoptosis, including intrinsic and extrinsic pathways, which involve various apoptotic genes, proteins, and signaling cascades. Dysregulation of this process can lead to renal dysfunction and contribute to the development of kidney diseases. Understanding the mechanisms underlying renal cell apoptosis is crucial for developing therapeutic interventions to protect renal cells from excessive cell death. Tubulointerstitial fibrosis is a common pathological feature of several kidney diseases, where excessive deposition of extracellular matrix proteins occurs in the renal tubules and interstitial space. This fibrotic process disrupts normal renal function and impairs tubular integrity. The cellular mechanisms involved in tubulointerstitial fibrosis include activation of fibroblasts, immune cell infiltration, and dysregulated matrix remodeling. Elucidating these cellular mechanisms is essential for developing targeted therapies to prevent or reverse tubulointerstitial fibrosis. The distal convoluted tubule, DCT, is a critical segment of the nephron responsible for fine-tuning sodium reabsorption. Na plus Cl cotransporters and mineralocorticoid receptor-mediated regulation play a crucial role in sodium transport across the DCT. Understanding these regulatory mechanisms is essential for maintaining overall sodium balance and blood pressure regulation. The collecting duct plays a pivotal role in concentrating urine by reabsorbing water and regulating the expression of key transporters, such as aquaporins. The molecular mechanisms underlying urine concentration involve the actions of antidiuretic hormone, ADH, and the urea transporter. Proper regulation of these mechanisms ensures the maintenance of body fluid homeostasis. Cell-cell junctions, such as tight junctions and adherens junctions, are crucial for maintaining the integrity and barrier function of renal epithelial cells. These junctions facilitate selective paracellular transport and play a pivotal role in preventing the leakage of solutes and water. Dysregulation of cell-cell junctions can lead to compromised renal filtration and contribute to the progression of renal diseases. Glomerular diseases encompass a group of renal disorders that primarily affect the glomeruli, the filtration units of the kidney. Understanding the cellular basis of these diseases involves unraveling the intricate interactions between glomerular endothelial cells, podocytes, and mesangial cells. Dysregulation of these cellular interactions can lead to glomerular dysfunction and the development of conditions such as glomerulonephritis and nephrotic syndrome. Podocytes are highly specialized cells that form the outer layer of the glomerular filtration barrier. These cells are crucial for maintaining the selective filtration capacity of the glomerulus. Podocyte biology involves intricate cellular processes, such as foot process dynamics, cytoskeletal organization, and signaling pathways. Understanding podocyte biology is vital for unraveling the pathogenesis of podocyte-associated renal diseases and developing novel therapeutic strategies. Chronic kidney disease, CKD, initiates a cascade of cellular adaptations aimed at maintaining renal function despite ongoing damage. These adaptations include hypertrophy, inflammation, oxidative stress, and fibrosis. Investigating these cellular adaptations can aid in the development of targeted therapies to halt or slow down CKD progression. Inflammation plays a crucial role in the pathogenesis of various renal diseases. The activation of inflammatory signaling pathways, such as NF-kappa B and MAP pathways, mediates the release of pro-inflammatory cytokines, recruitment of immune cells, and subsequent tissue damage. Understanding these pathways can provide insights into therapeutic interventions aimed at modulating renal inflammation. The reabsorption of phosphate in the kidneys is vital for maintaining systemic phosphate homeostasis. Several transporters and regulatory factors are involved in renal phosphate reabsorption. Disruptions in the molecular mechanism of phosphate handling can lead to disorders such as hypophosphatemia or hyperphosphatemia. Diabetic nephropathy is a leading cause of end-stage renal disease globally. A multitude of cellular and molecular events, including hyperglycemia, oxidative stress, inflammation, and fibrosis, contribute to the development and progression of diabetic nephropathy. Understanding these events is essential for devising effective therapeutic strategies to prevent or treat this debilitating kidney disease. The kidneys play a crucial role in regulating blood pressure through various mechanisms. These include the Renin-Angiotensin-Aldosterone system, tubuloglomerular feedback, and sodium handling in the distal nephron segments. A comprehensive understanding of renal contributions to blood pressure regulation is vital for the management of conditions such as hypertension. In conclusion, a deep understanding of renal cell biology is vital for comprehending the complex mechanisms underlying renal function and dysfunction. This comprehensive knowledge will aid in the development of targeted therapies aimed at preventing and treating various renal pathologies. By exploring the 12 key topics discussed in this lecture, we have gained valuable insights into the intricate cellular mechanisms that govern renal function. I hope this lecture has provided you with a solid foundation to further explore the fascinating field of renal cell biology. Thank you for your attention, and I look forward to our future discussions on this subject.